So in this portion of the PowerPoint, we're going to talk about enzymes' role in chemical reactions. So what drives a chemical reaction? If reactions are downhill, meaning exergonic, so for example, here we have a diagram of an exergonic reaction, and we can see that we have a little uphill portion and then downhill, and with that we get a net release of energy. So why don't reactions that have a net release of energy, meaning don't require a net input of energy, why don't they happen spontaneously, completely on their own? So for example, we have this molecule here of starch. So why is it that a starch molecule just doesn't break down on its own? And the reason for that is each one of these glucoses in the starch molecule are held together by strong covalent bonds. So these covalent bonds just won't break on their own. True that once the bond breaks, there is a net release of energy, but we actually have to put some energy in first, invest some energy, before we can get that energy out. So that's what this uphill portion of our energy hill or energy diagram is showing. We have initial input, and then we get that energy out plus a little extra. So we get a net release of energy. So breaking down large molecules requires an initial input of energy. So here we have a pile of logs, right? You've all lit a fire or campfire maybe once in your life. The logs are not just going to burst into flame on their own. You have to supply an initial source of energy, a match, to light that fire. And we call that initial input of energy the activation energy. Activation energy is talking about the amount of energy that we have to supply to activate or to get a reaction to happen. So large biomolecules such as starches, proteins, fats that you're consuming in your food, we have to supply actually an initial little input of energy to start to break those down so that we can get a net release of energy to run all the chemical reactions in your body. So too much activation energy for life. So normally to break down a molecule such as a starch, it takes actually a tremendous amount of energy to do that. So this activation hill, it's a pretty steep hill. So if we could find a way to lower the amount of initial investment of energy, that would make the reaction easier to happen. So activation energy, the amount of energy needed to destabilize the bonds and cause a molecule to fall apart, it takes quite a bit to break a covalent bond. So if we could find a way to lower this activation energy, this hill, it would be a good thing. It would mean that reactions could happen easier and we'd have to supply less energy for reactions to occur. So what's going to lower that energy hill? What's going to lower that amount of activation energy? So again, here's our So to break down this glucose, supply the activation energy. So it would be really nice if we had something that would actually help us to lower that activation energy. So to reduce that activation energy, we add catalysts. Specifically, the catalysts that we're going to use in living systems are enzymes. And what catalysts do, or enzymes do, is that they lower the amount of that initial input of energy needed to get the reaction going. It decreases our activation energy. So here is an uncatalyzed reaction, meaning without an enzyme. Look at how much energy is needed to put into the system to get the reaction going. When we add an enzyme, look at the activation energy. Far lower, which means less energy to gather to get the reaction going, meaning the reaction can happen faster. So here's a catalyzed reaction. Look at how much smaller that activation energy is. So what's a cell got to do to reduce its activation energy? Get help, get chemical help in the form of enzymes. Enzymes, the main purpose of an enzyme is to lower the activation energy, meaning the energy required to make a reaction go forward. So enzymes are known as biological catalysts. There are lots of catalysts. So flame is actually a catalyst. Heat is a catalyst. Platinum in some inorganic reactions can be a catalyst. Catalysts are referring to anything that will help facilitate a chemical reaction. In biological systems, meaning living organisms, the catalyst is almost always an enzyme. Enzymes are proteins, as we talked about before, and every once in a while there are some enzymes that are RNA. There are a few examples. They facilitate the chemical reactions. They increase the rate of reactions without being consumed, which is awesome because that means the enzyme can be used over and over and over again. It reduces the activation energy, but it doesn't change the amount of free energy released or required for the reaction to occur. The only thing an enzyme changes is the amount of startup capital, or the amount of startup energy needed to get the reaction going forward. So if we look here at our energy hill, we see that we have a lowered activation energy, but we still get the same net release of energy that we would with or without the enzyme. So required for most biological reactions. Just about every reaction that occurs in your body requires some kind of enzyme, and there's a different enzyme for each one of those reactions because enzymes are highly specific. So our body has thousands of different types of enzymes. There is an enzyme for every reaction. You know, they say there's an app for that. In terms of our biological systems, there's always going to be an enzyme for that. So enzyme vocabulary. We talked a little bit about this during our lab period, but here's our enzyme, which is not at all what an enzyme looks like. It's more of our cartoon depiction, but the purple guy down here that kind of looks like a cracked egg, this is our enzyme. And fitting into our enzyme, or the molecule that an enzyme reacts with, is called the substrate. So here in our diagram below, these two little the hexagon and a pentagon joined together. So this is a dimer of sugar, probably. This is our substrate. When the substrate binds to the enzyme, it forms something called an enzyme-substrate complex. A complex just means a combination of two things. And so the combination of the two things are the enzyme and the substrate. And then the product is what comes out of that reaction. So the separated molecules here, this would be our product. And notice we started with a larger molecule and we broke it into two smaller pieces. That would make this what type of reaction? A digestion reaction. So this would be digestion, catabolic, and because we're breaking something apart, catabolic reactions tend to be also exergonic. So there are our products. And then where the substrate fits into our enzyme, this little hole down here, that's known as the active site. That's where the substrate binds to our enzyme. So properties of enzymes. Properties are reaction specific, meaning that each enzyme works only with a specific substrate and does only one chemical reaction. They're not consumed in the reaction, meaning the enzyme can be used over and over and over again. And they're affected by cellular conditions. Any condition that affects protein structure, things like temperature, pH, salinity. This is why it's a big problem if you get a high fever. So if you get sick and you get a high fever, so looking at like 103, 104, 105, the problem with having such a high fever is it causes the enzymes of your body to start to do something called denature, lose its shape, and therefore the enzymes start to lose their function. And if you lose the function of the enzymes in your body, that can lead to severe health problems and even eventually death. So naming conventions. So enzymes, one way you can tell you're talking about an enzyme ends in ase, sucrase, protease, lipase, polymerase. Okay, and here's our exception to the rule because there always is pepsin. So these are all different enzymes, and usually they're named after the substrate that they break down. So sucrase is an enzyme that breaks up sucrose. Protease is an enzyme that breaks up protein. DNA polymerase. So you can actually see the word polymer, I hope. Polymer right there, polymerase. This is going to be an enzyme that builds a polymer of DNA. So one model or one hypothesis about enzymes is this idea of lock and key. So the lock would be the enzyme, and the opening in the lock is the active site, and the key would be the enzyme. So there's only one substrate, one key, that could fit the lock of the enzyme. A more accurate model of enzyme action is actually something called the induced fit model. So there's a little wiggle room to the shape of a protein. The protein is not necessarily, protein meaning enzyme, is not really rigid. The enzyme can actually slightly change shape, And so when the substrate comes in and attaches to the active site, the enzyme will actually change its shape to fit the to fit the substrate more perfectly. So the enzyme actually changes shape slightly, specifically at the active site, to more tightly fit around the substrate. So we call this a conformational change. Conformational is just a fancy way of saying shape. So conformational change, a change in shape. And it brings the chemical groups into the right position to catalyze the reaction. So either put stress on the bond to break the substrate, or to align two molecules at the proper angle to build a bond. So how does it work? There's a variety of mechanisms to lower activation energy and speed up reactions. So in synthesis, the way that activation energy is lowered is that the active site orients the substrates in the correct position to form the bond. Digestion reactions, the way that we lower that substrate and speed up the reaction is that the enzyme actually puts stress on the bonds that need to be broken and make it easier for the bonds within that molecule to be broken. And that's all I have to say about the introduction of enzymes and metabolism.